This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 233, recorded on December 31, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. Hello there. Happy New Year's Eve.
Yeah, indeed. And from some parts of Michigan, Michele Swanson.
Hello. Yeah, we call this up north.
Up north?
Up north, yeah. Wonderful to see you.
Now, if you keep going north, do you hit, this shows my poor geography, do you hit Canada eventually? Yeah.
You do, but so I'm in the lower peninsula, and then you've got to cross the Mackinac Bridge to get to the upper peninsula, where the Yoopers live, and then Canada.
Okay. And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Thank you all for doing this on a New Year's Eve. I appreciate it.
I was thinking about this crappy year that everyone has had, but you colleagues and friends like yourselves are part of have been bright spots. So I'm happy to see you today.
Oh, yeah, so true, so true.
I always say, I'm fine. I don't need to go out. I'm okay here. But it's because I have this community that we have built, right?
Right. Now we're very fortunate.
And we can chat periodically, and we can now see each other. And, we've been working together for a while, so we're friends.
Yes, we have.
Colleagues from all over the world are doing some really cool science that we get to share with our colleagues out there. So, it's a great episode today.
All right. If you enjoy our work here at TWiM and the other podcasts, you can support us financially, microbe.tv/contribute. And today, we as usual have a snippet in the paper, but our snippet is somewhat unusual, right, Elio?
Absolutely. Instead of discussing a paper, we're going to discuss a movie. And the movie is called Jezebel. And it's a movie from 1938 with no less than a very youthful, what's his name?
Henry Fonda.
Gosh. Henry Fonda and an equally youthful, what's her name?
Betty Davis.
Betty Davis. Okay, so it's Betty Davis and Henry Fonda, and they're playing in New Orleans in 1853, I believe. At the time when, and this is a love story. Okay, I won't go into the love story part, but I'll go into something else. The background is that that's the year when New Orleans was hit by a very, very bad epidemic of yellow fever. And the story here is a little bit analogous to what we go on through today with not an epidemic, but a pandemic. Theirs was an epidemic. There were several epidemics of yellow fever going on. It was a dreadful disease. It killed people. A lot of people died. And the story here is that the good people of New Orleans, thinking that the disease was transmitted by contagion, by touching people, by direct contact between person and person, they closed down the city. So much so that they shot people who wanted to leave New Orleans.
Yeah.
And and since they thought that the disease was caught by contagion, by touching, as opposed to miasma, which was the competing disease, which was bad air coming through, and that's how you caught diseases. So contagion was something to be avoided, and since the whatever it was, this is before Pasteur and Koch, so they didn't know much about germs, but they thought there was something in the air, and to avoid that, they would set off guns and cannons.
Absolutely cannons.
And great big bonfires in the middle of the street. And they were so afraid of people who got sick that they shipped them off to a Lazarette Island, an island which had previously been a leprosarium, that is a place where lepers went. So they shipped them off, and that's a good part of the story. Now, it took a while to find out that yellow fever virus is transmitted by insects, mainly by mosquitoes. This was a very big breakthrough. It was done by a Cuban by the name of Carlos Finlay, and then later on it was confirmed by Walter Reed, an Army doctor in Panama. And that was a huge breakthrough, because even though by that time germs were known, that is, the era of Pasteur and Koch was upon us, it still wasn't clear how bugs were transmitted. And the discovery that bugs were transmitted by insects was in itself a major, major contribution to science, to our knowledge of infectious diseases. So they had it all wrong. The people of New Orleans had it all wrong. They thought that this virus, or whatever it was found out later there was a virus, was transmitted by touching, by breathing, by stuff like that. The disease kept on going and was found, and it caused epidemics in various parts of the world. It still does. It's still present in parts of Brazil and parts of Africa. But luckily, there is a vaccine, and the vaccine is enormously powerful. It's a very powerful vaccine. It was discovered by Max Tyler at the Rockefeller Institute. About when, Vincent? Do you remember the year?
1947.
1947. So it came in late, but it's a very powerful vaccine, and it's used. In fact, I think at one point when I was going to go someplace in South America, I was asked to take the vaccine, the yellow fever vaccine, because I was going to travel to places where the disease was prevalent. So here you have it. You have a movie that deals in a very strong way with infectious diseases and with the subject of our podcast. By the way, it's very nice of Vincent to let me do this in a bacterial podcast, because normally this belongs to This Week in Virology, not This Week in Microbiology. So thank you very much.
We as you will see today, we often talk about viruses.
One thing I should tell you is that the hero of the story, at the end of the movie, you find out that he has the disease and is shipped off to this island, this special island. However, halfway through the movie, and it's done very casually, you see him swatting a mosquito. So you assume that is when he caught it.
That's where the contagion was.
That's where the contagion was. So the director of the movie, which was Wilder, very famous director, put that in there. And for most people, it wouldn't matter. But for me, it did.
Yeah.
So I know when he caught the disease.
Did you pick up in the very early part of the movie where Henry Fonda is talking to a group of bankers, and there's a bit player who is the physician on the bank's board, and the physician pounds his fist on the table telling, during the last outbreak of 1830, there were not enough able-bodied people to bury the dead. And they're talking about cleaning up the filth in New Orleans. So there are some important messages in this movie, but you just have to be enough of a microbiologist to pick up on the subtle hints that Billy Wilder, the director, offers to us. And it's just absolutely fascinating rewatching the movie, looking at it with a different lens.
That's right.
And it was interesting that they wove the tension between the business, the bank, and the public health crisis, because in these board meetings at the bank, the one advocate kept saying, we have got to, take care of this contagion. But
and so, I bet you dollars to donuts, there were many banks that had that same discussion this past year.
Right.
So this is a movie that it's quite a good movie, first of all. So it's worth watching, and it can be watched online. You can stream it somehow. But as Michael said, if you see it through microbiological eyes, you pick up a lot of little things in it.
If you watch it through feminist eyes, I will say that a lot of it is a little bit hard to take, and we're glad that there aren't as many parallels.
Oh, yes.
For sure.
You're right.
Well, Betty Davis did introduce the feminism in the very beginning of the movie when she's late to her own party, she's riding a horse, and she doesn't care about showing up in horse clothes, as they said. So it really established Betty Davis as that nonconformist.
Yeah, but she was also really bratty and selfish, and so she wasn't really held up as an icon. Yeah.
No.
So a few words about yellow fever. It was the first human virus identified in 1906, directly as a consequence of attempts to build the Panama Canal and many of the workers getting ill because the Aedes aegypti, the vector, was there. And Carlos Finley and others studied it. And in fact, the way they proved that the virus was transmitted by a mosquito was to have mosquitoes bite them. And a few of those individuals died as a consequence.
Wow.
But we didn't have a vaccine until 1947, and until then, mosquito control played a big part because in the Panama Canal, we recognized that it was Aedes aegypti. They're a tree hole breeder. They breed in small volumes of water, and all you have to do is control that, and you control the virus transmission. And that was effective until
you also control malaria at the same time.
Right.
Exactly. Although different mosquitoes, right? Anopheline mosquitoes. Anopheles. But the other thing, so the vaccine comes out in 1947, and now if you go to a place with yellow fever, like parts of South America, you should get the yellow fever vaccine. Here in the U.S., we had yellow fever outbreaks as far north as Philadelphia. There was a very serious one in Philadelphia. And,
wasn't there one in New York City also?
There's in New York City,
there wasn't one.
So the mosquito is no longer present that far north in the U.S. There's been a lot of mosquito control efforts. And in fact, New York City does scans for mosquitoes regularly, and they don't find any Aedes aegypti. They find other kinds of mosquitoes, of course. It's mostly limited to the southern parts of the U.S. Texas.
But is it creeping northward because of climate change?
Yeah, I mean, it will. It certainly will creep northward. But, 1 degree, which is a lot of climate change, is not enough to really impact it yet. But eventually, if we don't check climate change, yes, it will. So now there are some viruses that are transmitted by Aedes aegypti, for example, Zika virus and dengue viruses, as well as yellow fever virus. And occasionally there are little outbreaks in Florida, southern Florida and southern Texas, but they don't get very far. So dengue, there have been some dengue outbreaks in the south there. So for the most part, the Aedes aegypti is not an issue for transmission in the U.S. But yeah, again,
say something about the Flavoviridae, to which the yellow fever virus belongs. By the way, Flavoviridae, flavor means yellow.
Yellow, and right.
It's called that because it includes the agent of yellow fever.
Right. Yellow fever, of course, because the virus damages the liver and you become jaundiced and you look yellow, especially the whites of your eyes turn yellowish. So that's where yellow fever, and they named the virus family Flaviviridae after yellow fever virus. And there are many other members, dengue virus.
It's quite a group, isn't it?
Zika virus, and many, many other viruses, all mosquito-borne, many are mosquito-borne. And we just did a TWiM, actually, from the American Society for Tropical Medicine and Hygiene meeting, which, of course, was online this year. But we talked with 5 people who work on arboviruses, which are still very important. They've kind of been pushed to the side in this pandemic year, but quite important.
Anthropod-borne means anthropod-borne, which is
by
not
the way,
just mosquitoes, but it's
by the way, Elio, you said that this movie got a lot of things wrong. And I say that whenever they try to make a movie about infectious disease, or maybe even science in general, they often get it wrong. So Contagion is a movie about a pandemic, right? A new virus emerging from a zoonotic source and spreading around the world, making a vaccine, blah, blah, blah. Ian Lipkin, my colleague, was the script consultant. They still got things wrong. Okay, so it's hard to, for example, I'll just quickly tell you one example. So they're developing a vaccine for this new virus, right? And they're testing it in monkeys. And the lady doing the work, there's one woman, and you don't see anybody else except her. She injects one monkey with this candidate vaccine, and that monkey is protected. And then from that, they decide to immunize the world. Okay? They don't do any clinical trials. Here's the thing. So she takes this candidate vaccine, and this has been tested as an intranasal vaccine in the monkeys. She takes a syringe and she jams it into her thigh. She pulls it out, and then she goes to see her dad, who is sick, as a way of challenging herself the very same day.
So she forgot her basic immunology. She needs to take Brienne's course.
And then, of course, they end up giving this vaccine intranasally, not by injection, which is the way she tested it. So I asked Ian Lipkin, I said, why did you let them do that? And he said, well, what happens is you have a script and I look at it and I say, okay. Then the actors come and they look and they go, this is no good. We're going to change it. And they change it right on set. And so she said, I'm not going to just put this virus up my nose. I'm going to jam it into my thigh. It's more dramatic. And the director goes, well, Ian's not here. Sure, go ahead. I mean, these are expensive actors. You got to pay them a lot of money. The kind of prima donnas, right? So that's what happens. Science goes down the tube.
And not that many people watching are going to pick up on that distinction.
No, but I have to say, in my virology course, for extra credit every year, I say, watch Contagion and tell me 3 things that are wrong with it scientifically. And they love it. They love it.
Fun.
It's a fun exercise. And they always say, you know, I saw this movie before, but man, am I looking at it in a different light after taking this course.
Oh, yeah.
Oh, that's wonderful.
No, that's fun. Anyway, thank you, Elio. That was a lot of fun.
Jezebel has mainly one thing wrong. One thing is wrong.
Not bad. Well, of course, we didn't know much, so that's easier.
Sure.
All right.
Yeah, 1938, they didn't know much virology.
No, we didn't even know viruses existed.
1858.
The movie was made in 1938, so they couldn't fix what
we knew less.
Know.
Yeah,
that's right.
True. All right, I want to tell you about a paper, a quite interesting paper. It is published in Nature. The title is Prokaryotic Viperins Produce Diverse Antiviral Molecules from a group at the Weizmann Institute, including Bernheim, Millman, Ophir, Maitav, Avraham, Shomar, Rosenberg, Tal, Melamed, Amitai, and Sorek. And Viperins. Well, first of all, what does it stand for? It's a made-up human word. Viperin stands for virus inhibitory protein, endoplasmic reticulum associated, interferon inducible. Okay, that's where Viperin comes from.
See, I thought it had to do with vipers.
No, nothing to do with vipers.
Nothing to do with vipers.
It's a cool name, Viperin, and it's a member of this. So it was originally discovered as a human protein. It is induced in cells, or its transcription is induced by interferon. So when cells are infected with a virus, the cells sense infection in a number of ways, and within minutes, interferons are produced, and the interferons bind to receptors on cells, and they in turn turn on the transcription of defense genes. And there are over 1,000 such defense genes. They're called interferon-stimulated genes, or ISGs, and one of them is Viperin. So this was discovered a number of years ago. And in fact, back in 2018, I wrote a blog post, and the title, at Virology blog, the title is A Cell Protein That Synthesizes an Antiviral Ribonucleotide. I thought this was so cool, right?
You're right. It is cool.
So this is a cell protein that synthesizes an antiviral drug. All right. So it is actually has a very broad action. It inhibits a wide variety of RNA and DNA viruses, but in 2018, how it works was actually solved. And as I said, it's turned on by interferon. But when they cloned the gene, they saw that in the human genome, the Viprin gene sits right next to the gene encoding cytidylate monophosphate kinase 2. And if you treat cells with interferon, both Viprin and this CMPK2 gene transcription goes up. So they go up together. So this CMPK2 actually modifies a nucleotide. So they thought, well, maybe Viprin modifies a nucleotide as well. So what they did is they made the Viprin protein and they incubate the protein with nucleotides and deoxynucleotides, right? The precursors of RNA and DNA. And what they found was it converts CTP, cytidine triphosphate, right? Which is a building block of nucleic acid, to deoxy-3,4-didehydro-CTP. Now that is basically CTP without one of the hydroxyls on the ribose sugar, right? So CTP has a ribose sugar, it has a cytosine base, and then it has 3 phosphates. And below the phosphate, there are 2 hydroxyls on the bottom of the sugar ring, and one of those is missing in DDH-CTP.
And it's a really important one.
It's a very important one.
You get chain termination. That's the 3' hydroxyl. And when you add bases to DNA, that's where the next one is added on. And if you don't have a 3' hydroxyl, that's the end of the chain. And that's why we call it a chain terminator, right? So Viprin catalyzes the synthesis of this. That to me was just remarkable. We make our own antiviral drugs because a lot of antivirals that are licensed are chain terminators, like acyclovir for herpes viruses. It's a chain terminator. Right. Many anti-HIV drugs are chain terminators. And here we are, our own cells make one right in front of our eyes, and we never knew about it.
And it boggles the mind to think how that could have evolved.
Yes.
I wonder if you have any clues to share with us today.
Well, before you go and answer that question, this is how much of our CTP pool of a human cell is this molecule able to convert? Does it convert 10% of our pool?
I don't know. It's a good question.
It converts enough.
It converts enough to inhibit. Yeah.
There's enough to inhibit.
But the key is that the cell apparently is not damaged by this at all. So the idea has been emerging that cell polymerases are not inhibited, which is weird because you would think that without the hydroxyl, maybe they don't incorporate it, or maybe it's corrected, but there's no toxicity.
Or the virus is just simply using more of the pool, and it's consuming because it's using more of the pool at a faster rate.
It could very well be
consuming more of it. It's simply a rate issue
because the selectivity is not a property of all the chain terminating antiviral drugs. So AZT, the first HIV drug, had terribly toxic effects because it also inhibited cell polymerase. It was not very selective. It was the only thing we had, so it was licensed, but it was very toxic. And then subsequently better ones were developed. But how this is selective is not clear, and it may be a matter of the size of the pool and so forth. Anyway, so it's an antiviral protein in human cells, and it turns out that in this paper they start by saying it's some bacteria and archaea encode genes that have similarity to vertebrate viperins. So what are they doing there? And that's what this paper is about. So if you do a homology search in a database of 38,000 bacterial and archaeal genomes, you get 1,724 genes that appear to have some homology or are homologous to human viperin. And they could put these into various clusters. And then they try to know which are the ones that are actually viperin and which are genes that are involved in housekeeping activities, because it could be either one. So they made a very interesting decision. They said, defensive genes, if this is a defensive gene in archaea and bacteria, it's probably part of a cluster of other genes that are involved in defense. So they call defense islands, right?
Kind of a play on pathogenicity islands.
Exactly.
Like type 3 secretion, yeah.
So most of the Viprin genes didn't do that, but in one of the clusters, 60% of the gene were found next to CRISPR-Cas systems and restriction modification systems and other defense genes. So bingo, maybe Viprin is a defense gene, right? So that's where they start here. So what do you do next? You say, let's do an experiment. Let's do a functional experiment. And so they took 59 genes in all of these Viprins that they pulled out of bacteria and archaea. They put the genes, they cloned the genes into E. coli in the control of an inducible promoter so they can turn them on and off. And then they infect with a whole bunch of phages. And they use phages like P1, lambda, T7, MS2, Q beta, which are the RNA phages. And about half of the VIPs that they tested, Viprins, at 59, had activity. So what is the activity? You turn on the gene for the Viprin, and then you infect with a phage, and then you can either look at the cell death, or you can look at the production of phages. And they actually do plaque assays. It made me so happy to see them doing plaque assays, right?
Well, and I'd actually like to share, if I could, them in this case is the first author, Audie Burnham, and she initially tried to do these plaque assays with the E. coli now carrying these homologs of Viprin, and she tried several of them and got nothing. Was really frustrated, but she thought hard about this and then redesigned the E. coli strain, came up with a different E. coli strain that she thought would get around the problem. So she then repeated this plaque assay and had stacks and stacks of plates. So the next morning, after trying the new engineered strain, she went right to the incubator, pulled out all these plates, and was just thrilled. She's like, oh my goodness, she had that really deep sense of discovery. And she said she spent the rest of the morning sharing the plates, showing them to everybody else in the lab and sharing her excitement.
Nice. That's great.
Yeah, it's really a beautiful functional assay.
It's a beautiful thing about a plaque assay. You can take the plate and show people. Yeah. All right, so some of these Viprins were able to inhibit a variety of phages. And now these genes, the proteins encoded in them, have an active site which contains cysteine residues. So they can change those cysteine residues, and the Viprins lose their anti-phage activity. So it tells you the catalytic activity is probably important. They also did a cool experiment. They took the human Viprin gene and they put it in E. coli, and in fact, it protects against T7 infection. How about that? Isn't that amazing? So these Viprins are in.
Yeah, shows you whose side you're on.
It's evolutions. Evolution
whose
is.
Side are you on?
I'm not on anyone's side. I think it's an interesting experiment that tells you how conserved this mechanism is. Right.
That's right.
So these Viprins are in vertebrate cells, eukaryotic cells. They're in bacteria. They're in archaea. So phylogenetically distinct. And so they say, well, maybe this is an ancient gene, or maybe it was shuffled, throughout evolutionary history. So they try and make trees, phylogenetic trees. They have these genes are in 176 different species, 14 bacterial and archaeal phyla, and they make a phylogenetic tree. And they find these Viprins group into 7 clades that follow the phyletic grouping of the microorganisms, right?
Nice.
So all eukaryotic Viprins are a monophyletic clade, which means they have a single origin. And the eukaryotic Viprins are their closest common ancestor is an archaeal species, which we know those are very close, right? So they say the clear monophyletic organization of this Viprin clade and its position suggests that eukaryotic Viprin was acquired from prokaryotes via a single event in the ancient history of the eukaryotic lineage.
Yay.
I just love that.
One
right?
up for the microbes. Yeah.
Of course, they were here first. Yeah. They were here for a long time.
The one thing that when you look at their figure, the phylogenetic tree of the Viprin family, you see clade 2, which is mostly archaea and some cyanobacteria. Yeah. I would almost say, without going into the raw data, I don't know how close it is to the archaea, but I would have guessed cyanobacteria would be more likely because of the eukaryotes capturing the cyanobacters ability to photosynthesize and then effectively making the chloroplast and protecting the cyanobacteria from seawater phages, which are quite prevalent. Remember, the phage concentration in seawater is about 1 million per mil.
Yeah.
And so they would need a defense mechanism like that. And so I when I was reading that, I said, I wish I could go and look at all the raw data, but I didn't have time to look at all the supplements to see to show how closely linked they were to the archaea.
That beautiful bioinformatic analysis was done by A.D. Millman, with a second author on this paper. And the first author wrote and said, really, without A.D.'s expertise, this project never would have gotten off the ground. It was really her ability to see patterns and make coherent arguments based on the phylogeny that really motivated them to carry on.
So I remember I told you that in eukaryotes, the Viprin gene is right next to another gene that modifies nucleotides. So they said, do we see this in our cases in bacteria and archaea? And in fact, 47 of 250 prokaryotic viperins were adjacent to genes that are annotated as a nucleotide kinase. Right, so it looks like a nucleotide kinase, not necessarily any functional work. And in some cases, this is very cool. The kinase gene was actually fused to the Viprin gene. So they're both produced from a single gene. Really interesting. Yeah. So they say that's more evidence that the Viprin is the substrate for Viprin. Because remember, we don't know yet in prokaryotes and archaea whether the substrate is a nucleotide yet. We're going to find out. But interestingly,
it's certainly consistent with the spread of this module, this defense module throughout. Yeah. Archaea bacteria, because you didn't need a whole pathway with dozens of genes, just these two.
That's right.
And the other thing that our listeners need to appreciate is bacteria. One of the hallmarks of bacteria transcription is polycistronic messages, whereas one message can produce multiple proteins.
Right.
And that's, again, a hallmark that then would get sliced and diced as it goes eukaryotic, if you will.
Yeah, that's right. You make one message and you can get multiple proteins, right?
On the theme of making funny nucleotides, remember we discussed once a paper where they found that Pseudomonas has the ability to convert ATP into tetraphosphate and pentaphosphate. So this is, and when that happens, some Pseudomonas can inject via the type 6 secretion system. It can inject this enzyme into other unsuspecting bacteria. And when they do that, they run out of ATP.
Right.
Because all the ATP gets converted into tetra and pentaphosphate. So this is sort of unusual. This adds to this excitement about the fact that there is a circus of nucleotide players, which is not just the way the ATGC that we loved and get used to it.
But they haven't shown that yet. They've just got the homology, and it's a strong prediction.
So far, the homology and the enzymatic activity, the catalytic site seems to be important. So, by the way, when they're looking at these genes, as I said, often next to the Viprin gene, there's a nucleotide kinase. In some cases, it's a cytidylate kinase, which is the same as in vertebrates. But sometimes there's thymidylate or other kinases. So maybe other nucleotides other than CTP are modified. So we're gonna get to that. But if you just look at the genes, it suggests that. All right. So next, they take bacteria, E. coli, producing these Viprins, and they extract.
By the way, you're saying Viperin, not Zipperin. It comes through as Zipperin on this audio.
No, it's not Zipperin, no. It's Viperin. V as in Vincent.
Right.
Or Viper, whatever you want. So they produce these, they express the genes in E. coli, and they extract small molecules. They say, whatever is being modified must be in a small molecule fragment. They do mass spectrometry, and the positive control are lysates making the human Viprin protein. And when you look at the positive control, you get a compound that looks like DDH-CTP, and they can further identify it by mass spectrometry. So human viperin produced in E. coli makes DDH-CTP from CTP. And then they look at the E. coli making all the 27 different viperins, which were anti-phage, had anti-phage activity. And they identified derivatives of CTP, including DDH-CTP by mass spectrometry, and so showing that, in fact, these viperins can generate DDH-CTP.
And let's just go behind the curtain and back into the lab the day that they got these results. While they were envisioning this process, she was presenting to her lab and wrote like, okay, here's the result that would be too good to be true. And that is if we could demonstrate that they do produce novel modified nucleotides. So they set up to do the experiment, and the day she got the mass spec and looked at them just for a few minutes, she got like just so excited. She went looking for her mentor, and this is Rotem Sorek. Wasn't in his office, so she texted him, and within 15 minutes, the two of them were poring over the results, and they were just in awe at the diversity that they found in these mass spec. And they said even in their craziest, wildest dreams, they did not expect such beautiful results. And some of the things they predicted, and then other things they hadn't yet found the activity, but they know that they're antiviral, so there's yet more to be discovered. So she said that was just like such a highlight to share that thrill of discovery with her research mentor.
And aside from the cool science that they did, each one of these novel nucleotides that they are identifying from these viperins are actually potential drugs that we can be developed.
That's right. That's right. So bacteria not only give you antibiotics, they can give you antivirals, right?
Yeah.
That's pretty cool. All right. So they found that some of these VIPs could make DDHC, but others didn't make it. So they couldn't find anything that looked like DDH. So they said, what else is it? And they found DDH GMP, DDH UTP. So it modified other bases to form the DDH derivative. So we have CTP, GTP, and UTP are modified by these different viperins. So some will modify C, some will modify G, and some modify U, which is very cool. So the eukaryotes got one version, but they have more, which are more diverse, I guess, if you want to say. So also an interesting aspect is that sometimes the viperins make one modified nucleotide. Some of them, 11 VIPs, could make multiple DDH ribonucleotides. And for 5 of them, they didn't detect any modified nucleotide. So maybe they make something else that, wasn't picked up in their mass spec protocol. Isn't that cool? So there's a lot more to be found. So far, we talked about ribonucleotides. So that would protect against RNA, but the viperins also protect against double-stranded DNA phages. And so they think that it's actually transcription that's being targeted in those viruses, not DNA replication. And in fact, in mammalian cells, it had been shown previously that viperin will inhibit transcription. So they did an experiment to address that. They took T7 RNA polymerase. They put a plasmid in E. coli that contains the green fluorescent protein gene driven by a T7 promoter. And boy, folks, parenthetically, I got to tell you, T7 promoter, that's how they're making the mRNA vaccines against SARS-CoV-2. But in vitro transcription. And they can induce the synthesis and see a fluorescent GFP signal. And when they put viperin, either human or the bacterial or archaeal viperins in there, it represses the GFP signal. So it's it doesn't show it's at the level of transcription because GFP has to be translated first, right? But it's suggesting that it's transcription because there's no DNA replication in this system. But then they go on and they extract RNA from these cells and they sequence it. My gosh, they do deep sequencing of RNA-seq of this to prove that the GFP gene transcription is actually being inhibited by these chain terminators. I was thinking today, what would I have done in the old days? Well, I wouldn't have done RNA-seq because I didn't have RNA-seq. Probably would have done a northern blot or something like
Northern
that.
blot to look for a smaller message.
Yeah, but they have the technology and they can show that. So their idea is that these viprins defend against phage infection by suppressing transcription. Now, T7 encodes its own viral RNA polymerase, right? And those are inhibited by these terminators. And by the way, these viprins are not toxic to E. coli, just like mammalian viprins are not toxic to mammalian cells. So some of the phages they used, which are inhibited by viprins, don't encode their own RNA polymerase. So they don't know how they're inhibited by the viperins because that's a host polymerase, right?
Do you think it's playing with sigma? Because a lot of the phage encode a novel sigma factor, so it only reads the phage promoters.
That would be a different mechanism, right? Yeah.
It'd be a different mechanism, but it's still inhibiting the RNA polymerase. But remember, the RNA polymerase is a multi-protein enzyme complex, and sigma factor is what confers promoter specificity.
Yeah, they do say that it's some mechanism independent of these modified nucleotides. So it could be sigma or it could be something else. So more to do,
More to do.
Never finished. One more interesting bit of work. They remember these viprin genes are part of what did they call it? A defense island, right? There are other genes in there. And they said most commonly they found two other genes in addition to the viprin gene. One of the genes encodes an ankyrin repeat domain protein, and the other gene encoding an RNase. Now, ankyrin repeats are motifs that are often involved in protein interactions, and they say maybe this is a sensor for the phage. Maybe this ankyrin repeat protein somehow senses infection and activates the transcription of the viprin gene. And then the RNase may also be involved in degrading incoming phage RNAs as well.
So ankyrin is the prokaryotic equivalent of interferon.
Could be. Well, if it's turning on the transcription, yeah, it would be.
Yeah.
Right. Seems to me they're suggesting that it's both sensing and turning on, which could be, could be a simplified
multifunctional protein.
Yeah.
A multifunctional protein.
So that's the story which I just found remarkable. I've followed viperins in vertebrate cells for a while, but now they say this is another strategy of chemical defense against phages, and they say maybe small molecule inhibitors are more common than we previously thought. Right, so these are nucleotides, and that's a small molecule, and we definitely have not thought much about those being common, and they suggest at the end, these could be adopted to antiviral drugs.
Yeah, and to mine the microbial world for not just antibacterials, but antivirals. Yeah. And in fact, a number of the authors on the paper are with Pantheon Biosciences, and they contributed a lot of the biochemistry expertise. And the plan is that they are now going to develop this project in the private sector. Cool. So if I can share, again, Adi Burnham, the first author, wanted to credit the head of the lab, Rotem Sorik. She said that he really fueled the project because he has a lab that has a very collaborative and creative spirit that really drove the whole project. And then I've already mentioned the...
Almost every paper from that lab is exciting.
Is that right? Wow.
Absolutely.
And then I already mentioned the bioinformatics by Adi Millman, but she also wanted to credit Helena Shomar, who is now with Pantheon Biosciences and did some of the deep biochemistry that we just learned about. And it turned out that Adi and Helena were friends from back when they were undergraduates and had many really productive conversations over the years. Adi learned to turn to Helena when she had a technical biochemical problem. And after they had a visit and a vacation together in Israel, Helena decided she wanted to join this project through Pantheon Biosciences. But let me tell you about Audie's path to a career in science. She never thought, as when she was a kid or a teenager, this is what she'd pursue. In fact, as a college student, she studied biology, chemistry, math, but also economics, agricultural development, and public policy. And then she went on and got masters both in biology and then a second one in public policy. So she really is committed to working on behalf of society and doing outreach. And in fact, one of the papers that she published for her master's was quantitative evaluation of public policies for gender equality. And she's done quite a lot of outreach work, including a consulting mission with the French Ministry of Women's Rights, looking at the interface of research and public policy on gender equity. And I direct you to her website where she describes a lot of her work. So she's committed to making society better. But she said once she got into the lab, she really caught the bug, was so excited by it that she committed and can't imagine doing anything else. So she ended up getting her PhD at the Pasteur Institute, and then her postdoc was at the Weizmann Institute in Israel, and that's the work we just learned about. Currently, she's at the INSERM in Paris, an institute in Paris where she's with the Infection Antimicrobials Modeling Evolution Group. She would like to also share that when she thinks about how she would advise junior colleagues or aspiring scientists, she says, it's a really tough, very personal decision. But what's worked for her is to make career choices based on what really fascinates and motivates her rather than what people might expect or what would look good on her resume. And she said that although working on both public policy and biology was not conventional, in the end, it's worked out really well for her. She also can recommend that people choose labs not based on a particular scientific question, but also really look at the team and what is the culture of the lab. So she's always looked for labs that are not only doing excellent science, but also have generous, creative people. Because she says, I can be interested in lots of different amazing biology, but it's really important that I'm part of a team that's collaborative. And again, she credits Sorek for setting up that culture in the group. So that is her story, and I look forward to her contributions in the future.
Nice. Thank you, Michele.
Yeah.
It's a lovely piece of work. It's just
it really is
beautiful how it progresses logically and everything makes sense. There are things to be discovered, and I really appreciate it because I have to tell you, there's a sea of COVID-19 stuff that is frankly not very interesting out there because it's not done very well. It's rushed. And I don't get it. Well, I do get it. People are trying to rush and get grant money and so forth. But really, folks, all that matters right now is getting this vaccine out there. The rest is. But there's a lot of fundamental stuff to be discovered. But this is beautiful.
Well, that's one of the highlights of this paper is, for the last 20 years, the pharmaceutical industry has been screening libraries. Mm-hmm. And they didn't really discover the viperins that the prokaryotic world has to offer in the screen of their libraries. So we really need to ask our friends who are in the screening business, why or how did you miss this family of remarkable proteins that the prokaryotic world seems to have adopted wholesale? I mean, there's quite a number of them. They're part of this defense island. And remember, the screening libraries also didn't tumble into the whole issue of CRISPR-Cas. Yeah. So there's something to be said for how these authors approached this story. And as Vincent led us off with, he wrote about this over 2 years ago in his blog post about what the viperins were doing in the eukaryotic world. But the bigger news is what the viperins are doing in the prokaryotic world.
It's amazing. But all these genomes are sequenced. It's just a matter of looking at them. And I think the bottom line here is that there are too many genes and you need to have some focused approach. Hey, let's take this eukaryotic protein and see if it exists in other worlds. And that's what they did.
Yeah. And then to do the functional assays and then the beautiful phylogenetics and the biochemistry. It really is
the assays are all beautiful. It's just a great combination. It's what I really like to see. So congratulations to everybody on that paper. And,
absolutely.
Yeah, beautiful. I'm going to write another blog post about it because it deserves one.
Well, it's a great way to celebrate the new year.
Yeah. All right, we have one email for you before we say goodbye. This is from Marco, who says, "Hello, TWiM team. I'm just another microbe lover like you, and I wanted to spread this wonderful footage of actinobolina hunting and catching its prey. Actinobolina. Elio, what's an actinobolina?"
I'm sorry. It's above my pay grade.
Actinobolina is a ciliate. Okay. It's a ciliate. Yeah, so it is above your pay grade because it's a carnivore.
Well, in the post that is on YouTube, which I commend the audience to watch, it's a tiny predatory porcupine is how they entitle it. And if you look at the video, it is so cool because what this little tentacle-laden ciliate does is it stings its prey and then it eats it. And they have the most incredible video illustrating this feeding process. You can witness it at about, you see the cute little ciliates floating around in this algal mat, but the video that he offers us is just absolutely incredible. And if you want something to do in between the parade tomorrow or the parade will be over by the time this gets posted, but take a look at this and it's really quite fascinating.
So Marco continues, you could perhaps check out some other videos by this YouTube channel they named Journey to the Microcosmos, as it would fit your interests. They've been doing these for quite some time. I thought I'd spread the word to you and your listeners. They have a wonderful collection of the microscopic world that surrounds us. And on occasion, there's a lovely story attached to the video for your listening pleasure. This one was quite fun and interesting. And Marco gives the link to this actinobolina. Thank you, Marco. And that'll do it for TWiM 233. That's also it for TWiM in 2020. No more 2020 episodes. And when you hear this, it'll be 2021. Good?
I say goodbye to 2020 is a pleasure.
Yeah, it's true. It's not been a great year, that's for sure.
It sure hasn't.
You can find TWiM at microbe.tv/TWiM, where you'll find the show notes. If you want to send us questions or comments, TWiM at microbe.tv. And if you like what we do, support us financially, microbe.tv/contribute. Michele Swanson's at the University of Michigan in Ann Arbor. Thank you, Michele.
Thank you. And Vincent, congratulations on episode 700 for This Week in Virology. Thank you for all you do.
Thank you. Thank you very much. Appreciate it. Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure. And again, Happy New Year.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone. And Vincent, happy birthday on Saturday.
Thank you very much.
Oh, my.
That's right, January 2nd. I'm Vincent Racaniello, and you can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Happy New Year, everybody. Thanks for listening. We'll see you next time on This Week in Microbiology.